Well, good morning, everyone. Thanks for coming again today. And hello, everyone downtown. I have a couple of announcements first, and then we'll go on with Lecture 15. First, I'm wearing the last year's ExxonMobil Texas Science and Engineering Fair. I invited you all to volunteer to help us out. I appreciate those of you that, or my appreciation goes out to those of you that have signed up already. If you're not able to volunteer to help us, I hope you pass it on to fellow university colleagues and encourage them to help out. It's a big event. We need a lot of assistance on that, and we really treasure your contributions. Question, yes? It should start probably next week. I'll be meeting with the volunteer coordinator either today or tomorrow, and we'll get that initiated. The contacts will be both through email and phone. Okay, the email that you put on the website when you registered. It's the setup at the convention center, and where we need assistance to pack things at UTSA and get them down to the convention center, and then ultimately to get them into the convention center and set them up, is Wednesday, March 30th. Thursday, March 31st is when about 1,000 pre-college students will be arriving, their teachers and parents and everything from all over the state, and they register and they set up their exhibits, and there's a bunch of tasks to do on that day. The Friday is the judging in the morning, and then doing various nice things for the students once they leave, and we can get in there and put treats at all their 1,000 posters. So one person distributing 1,000 ribbons is quite a task, so there's things like that we need help with. And the Saturday in the morning is the award ceremony, so setting up for the award ceremony is quite critical for Friday night as well. So April 2nd is the last day, that Saturday, and after the award ceremony and everybody, all the students take down their poster exhibits and clear out, then it's time for us to clear out our stuff as well and make room for the convention center to do what they need to do. So that's kind of it in a nutshell, but there's a lot of subtle details, badge checkers and keeping the traffic flow going and stuff like that. So brave people that will tell someone, no, you got to go park first, go get your free parking, then walk over, and then go back to your vehicle, and then come back again. Otherwise, we'll have the off-duty police department officers just telling people, no, you can't turn in, you got to keep the traffic moving. So there's a lot of tasks like that. I've forgotten the second thing. Oh, the second thing is I have to attend a meeting of the Texas Science Careers Consortium on Friday. I think I sent out an email, so I will not be in class on Friday. I remind you that Lecture 15 for today, there's a 1 and a 2 from last fall that I've already changed the name on, and there's a 15-2 pre-recorded streaming video for this lecture, and then Friday's scheduled lecture, Lecture 16, is also recorded from the fall 2003 semester. So I'll rely on you to turn to those resources to cover what information we might not get in today and Friday, and then on the following Monday we'll be back on schedule with Lecture 17. So let's switch over to the computer and discuss the body cavities. Again, we're really considering this overall morphogenesis of the embryo at this time, and we've talked repeatedly about rapidly dividing cells, migration of cells, either a plate-like action or just bulk movement, or other types of cytoskeletal dependent motility, such as the mesenchymal cells kind of migrating around, and we'll see later other cells such as the germ cell primordial cells coming from the yolk sac and navigating into the abdominal cavity. Those obviously require some motility of the cells, an interaction between the glycocalyx or the glycoproteins on the surface of the cells interacting with the extracellular matrix proteins to navigate. So there's a lot of changes in all the chemistry and gene expression as these events are occurring. But most importantly, what we want to establish now is just a general feeling for how the major and minor body compartments are developed. We will be focusing today primarily on the ventral body cavity that's divided into the pleural cavity where the lungs are located, the pericardial cavity where the heart is located obviously, and the peritoneal cavity where the abdominal pelvic structures are surrounding it at least. We need to remind ourselves of these potential anatomical spaces that we're considering are actually fluid-filled spaces. They're not of tremendous dimensions. The layers that create the lining of this enclosed cavity, somewhat like the amniotic cavity, have a simple squamous epithelium, and they are typically in contact with each other in the fully developed organism. There's a larger fluid cavity for what we're considering now. Remember, amniotic fluid is basically seeping through everything. It's permeating throughout various origins and circulations. So these are larger dimension body cavities that we're seeing in the slides upcoming. But ultimately, those body cavities really become very narrow, and there's that thin film of serous fluid secreted by the lining membranes, the serous membranes, to reduce the friction and allow the lungs to move, the heart to beat, and the intestines to jiggle around when we do things. So we're focusing on this global formation of a three-dimensional organism. We went from the 2-cell layer preembryonic disc to the 3-cell layer preembryonic disc. We started to get folds and bulges and movement of structures to see the first signs of organogenesis. The cardiogenic region way out in front of the precordial plate ultimately is going to fold and come directly into the chest to put the heart in our chest and the diaphragm to develop inferior to it. So they start off with different relationships, and as they're folding down, those relationships change with reference to each other. And obviously the nervous system beginning to develop as well. So these are the first signs of organogenesis that we've seen to date. In addition to this, with the circumferential growth and development, the plate-like movement of structures, and still that constriction of the junction of the amniotic sac and the yolk sac, there has to be folding that is going to occur. And this was discussed in a previous lecture, and it'll come back with some indications of how these body cavities form. So there's lateral folding. We talked about envisioning the butterfly wings coming together and fusing in the front. That'd be the lateral folding. And then there's going to be a head fold and a tail fold to form the C-shaped organism, the embryo, ultimately from the crown to rump dimension along the long axis of the surface of the back with the reference to the C-shaped appearance. So we remind ourselves of intraembryonic coelom. There's an extraembryonic coelom. The coelom is referring just to this fluid-filled space. So it is a cavity. One of the first ones we saw in the concepsus was what? What was the first fluid-filled cavity that we saw in the concepsus? The blastocyst, that's right, the blastocystic cavity. Well, now we have the pericardial cavity that formed basically from the U-shaped intraembryonic coelom that we illustrated in a prior slide on a dorsal view. And the pericardio-peritoneal cavities we mentioned are established by that coelom forming in the intermediate mesoderm. And the pericardio-peritoneal cavities will contribute to the pleural cavities for that portion that's in the chest region, basically, the primordial chest region, and lateral to the foregut and dorsal to the septum transversum that's going to become the diaphragm. So there's several structures that are going to move into place and subdivide this pericardial peritoneal cavity and leave behind basically the space into which the lung buds are going to grow. So we now begin to envision these cellular growths from the primordial germ cell populations, and they're going to invade certain areas, and as they push in, they're going to have on their innermost surface, with reference to being inside the body, outermost with reference to the cord of cells that's growing in, or a tube of cells growing in, and that's going to be lined by that simple squamous serous membrane lining the organ, and then the rest of that serous membrane surrounding the pericardio-peritoneal cavity is going to be the same layer. It's a continuous layer, let me rephrase that, and it'll be plastered against the body wall. So for those of you that have taken anatomy courses, you know that this layer of serous membrane lining the inner body cavities that's actually on the body wall is called the, what is it called? The parietal layer. So it's on the parieta, or the walls, and then that ends up on, say, the lung buds growing in or the heart is going to be the visceral layer. So we're going to be talking about a visceral layer and a parietal layer of all of these serous membranes, initially continuous, forming the pericardio-peritoneal cavity, that U-shaped structure, and as it's partitioned out, it'll be parietal and visceral pleura, parietal and visceral pericardium, and parietal and visceral peritoneum in the covering the associated with the lungs, the heart, and the intestines, respectively. And obviously, the dorsal septum transversum with other structures will form the separation of the thoracic cavity from the abdominal pelvic cavity. So we are seeing the early signs of the formation of the complete three-dimensional, non-segmental organism. So the somites represented segmental development, and differential gene expression at different times disrupts this segmental development so that our organism, the human organism, is not segmental. So there's a peritoneal cavity, as we said, that will end up in the caudalmost portion of this complete pericardio-peritoneal cavity. Thus, we can take a quick look at this slide. We've jumped ahead. We see there's a lot of organogenesis established here. Obviously, the nervous system, the cutaneous system. The surface ectoderm has enclosed nearly all of the structures at this time. And this is in the 5th week. The circulatory system with the heart and several vessels is prominent. The digestive system, the digestive tube, is prominent. We see right here there's cutaneous membrane covering this opening into the gut tube, primitive gut tube. What was that? What was that little spot that was one of our initial placeholders, landmarks? The precordial plate. Remember, the precordial plate becomes the membrane covering the opening into the oral cavity, and actually this would be the first part of the pharynx right here. Other growth structures in this area are going to create the oral cavity. So it's that buccopharyngeal membrane, bucco referring to oral cavity, and pharyngeal referring to this first part of the gut tube. So that we see the digestive system is developing, along with some of the accessory structures such as the liver. So this is quite advanced in considering these initial fold, lateral and rostral and caudal folds. We can see the rostral fold, the forebrain pushing forward to create a curl in the front end, and the lengthening of the fetus with the addition of the somites creating the tail curl, or the caudal curve and fold, and then the obvious lateral folds, those butterfly wings, have come around and for the most part have met in the ventral portion, and we see that it begins to pinch off at a certain area that includes the allantoic stalk and a portion of the gut tube that feeds into the what's the other one here? The yolk sac, right? So the yolk sac is going to be here, and then the vasculature. So this obviously is going to become, it is the body stalk, and it's going to become the umbilical cord. So all of these structures are present, and we see the septum transversum now inferior to the heart and in place to separate the thoracic from the abdominal pelvic cavities, and we see some of the early signs of outgrowths of these endodermal layers, these cores of cells that are going to grow out and form the lungs and the other structures in these areas. So we'll take a look at some different planes of sections to try to recognize some of the adult body cavities at this stage in the 5th week. So first we're going to look at the section plane in this plane shown here for panel B. So we're going to see at about this level of this most rostral somite going through the esophageal area. Basically, this is the larynx is going to form very close to this area and be just in front of the heart. So what we begin with on these types of transverse sections is let's find our landmarks and figure out what we're looking at. So the first landmark is always going to be the neural tube. That tells us dorsal. And it's always easy to distinguish in any illustration or even an actual histological section through these areas throughout the entire extent of the embryo. Basically, we either have the neural tube or the forebrain structures that are our orientation for dorsal. So that means this is dorsal, this is ventral, and then we have the two lateral sides. The standard, basically, as we know, with clinical anatomical viewing is we're looking from below. So we take this section from the embryo with the head over here and turn it away from us, and we're looking from below, so right should be right and left should be left, correct? So this should be right and that should be left. And that's not a great concern for us now because we're not going to focus on that much, but as we begin to look at transverse sections where the liver is going to be displaced to one side and the stomach's going to be displaced to another, distinguishing right and left will be important. So we come back to our initial landmark. We remember that immediately ventral to the developing central nervous system is the notochord. So the notochord is going to persist. Remember that. It persists into adulthood as the nucleus pulposus of the intervertebral disc. Then we know that, we know that the spinal column is going to form from the paraxial mesoderm in this area. If the notochord is going to end up contained within the intervertebral discs, then the vertebral disc and the vertebral bodies are going to develop here in front of the spinal cord at this level of section. So just in front of the vertebral bodies, we know in the adult organism is the aorta, and then the vena cava is going to be there to some degree or another. It's a little more variable depending upon where the section is, but the aorta is going to be in front of the vertebral bodies, basically from the thoracic region, as we know, a little bit lower in the thoracic region, all the way down to the lumbosacral junction. So as we're proceeding rostrally from the initial landmark, we know that the gut tube is going to be in front of that. And we know that this vasculature is going to, this large vasculature is going to provide the smaller vasculature, the arteries and arterioles, to the gut tube, as well as the veins and the venules and the capillary beds within. So that vasculature develops very early on, and we saw that indicated in here. They're at least showing the midgut artery called the superior mesenteric, and here we can see for the stomach arterial branches already well established. So this obviously is not a complete illustration. There's a lot more vasculature present. But it's important to remind ourselves of the proximity of these two. It's either going to be very close, or it'll get stretched and be a little bit farther away. Here we are, obviously, in the thorax, because we know that the heart is present. Again, this is the 5th week, so it's flipped from being rostral, the most rostral structure, to being placed within the primordial thoracic cavity. So we know that the portion of the gut tube in the thorax is the esophagus, and we know that the trachea is going to be there. So we have this plane of section in this panel B right through the larynx, basically, where the trachea is going to be budding off, and the esophagus would persist in its connection on down through. So if the trachea is developing here, we know that the lungs are going to be developing nearby. If this is the thoracic cavity, there must be this serous cavity developing nearby as well, this fluid-filled cavity. So remember that the thoracic cavity and the abdominal pelvic cavity generate basically the ventral body cavity, divided by, of course, the thoracoabdominal diaphragm. And there's an inner lining of dense, not quite so dense, but irregular connective tissue called the endothoracic fascia and the endo-abdominal fascia. And especially it's well developed in the thoracic region, the endothoracic fascia, and that's really what creates this airtight container of the thorax. And then the visceral layer, the parietal layer of the pleura in the thoracic cavity is tightly adherent to that endothoracic fascia. So actually, if you pierce the endothoracic fascia with a structure, you're automatically going to go right through this simple squamous epithelium, the parietal layer of the pleura, and cause a pneumothorax, right? So when I say the thoracic cavity, we always think of these serous cavities contained within that. So it's a balloon within a balloon, if you want to think of it that way. So we know that this should be part of that U-shaped structure, the pericardio-peritoneal cavity that we recall was a simple U-shaped structure when we looked dorsally on the earlier embryo when it first formed. So if you can picture taking a U-shaped garden hose and then taking the front part of it and pulling it down in underneath, this is what happens to the pericardio-peritoneal canal. So the pericardial portion ends up down in the chest cavity, and then it arches up and around and continues back down into the abdominal cavity. So in this case, where we've got that loop bent around, we make a cut in the front part after it's bent, and we see the original mid portion of the pericardio-peritoneal canal, and then we get down into the curve of this horseshoe-shaped structure, and we cut through that as well in the more ventral location, and that's the pericardial portion of this. So we see in this plane of section two aspects of this continuous body cavity, the pericardio-peritoneal canal. Okay, so that it's obviously named for its site of origin and termination, and kind of disregards the in-between locations within the thorax. So basically, when this curving occurs, and the heart is evident, then we refer to this portion with the heart as the pericardial cavity, the caudalmost portion as the peritoneal cavity, and then the in-between as the pericardio-peritoneal canal that ultimately is going to become the pleural cavity. So as we continue forward then, we envision the development of the pericardium, the sac surrounding the heart. So this layer reflecting off the attachment, where the layers are continuous, goes on to the body wall. So that's going to become the the parietal pericardium, and the portion that remains on the surface of the heart is the visceral pericardium. The point of continuity of the parietal and the visceral mesothelial layers, mesothelium is another term for this serous membrane, simple squamous, is called the mesentery. So the mesentery is this attachment. We'll see that for the digestive system there's an attachment in the dorsal and the ventral aspect. For the heart there's just a dorsal connection. May not have been dorsal originally, it was probably more caudal, but as it folds under like this, it is a dorsal mesocardium. That obviously is not going to persist. The only attachments of the heart to the rest of the connective tissue structures in this space between the ultimate pleural cavity and the pericardial cavity is just going to be this loose connective tissue, the adventitia of many structures in this area. So this dorsal mesocardium will not persist. So obviously now we're starting to talk about structures that are going to appear and disappear, and there'll be persistent dorsal mesentery and persistent ventral mesentery in locations where they disappear ultimately, and things like that. So the discussions of these anatomical structures becomes more and more complex as we proceed. So the single tube-shaped structure of the heart is evident at this stage of development. And all this, the pink-colored collections of cells and extracellular matrix is really mesodermal origin stuff, and typically referred to as mesenchyme at this point. Now at level C, we are obviously somewhere near the origin of the stomach, so we are already caudal to the septum transversum, where which contributes to the formation of the thoracoabdominal diaphragm. So we find our landmark dorsally, the neural tube, the notochord immediately ventral to it, the aorta immediately ventral to that, with a branch coming off, the foregut artery called the celiac artery. Ultimately it would be called the celiac trunk because it stays rather short. And it's going to give the arterial supply to the foregut structures of the gastrointestinal system. Branches of this ultimately will get over and form the hepatic artery as well. The adult organization and collections of branches of this arterial and the venous system is very complex. You spend hours and hours trying to learn all the branches just of this one celiac trunk and to envision their distributions. So the gut tube is a bit more dilated here. The fact that it's located close to the liver. There's only two bulges, the cardiac bulge and the hepatic bulge, so you're either looking at heart or liver, and this is a solid structure, so it can't be the heart, it's got to be the liver. So we know we're in the abdominal cavity, the upper abdominal cavity. So this would be the dorsal mesentery of the foregut. And now we're seeing that there was a ventral mesentery here as well, and the liver has grown into that. Obviously, when we do organogenesis, we're going to see all this stuff again. So it'll come back for reinforcement and review at a later time. So there's a dorsal mesentery and a ventral mesentery, and we can already see that the dorsal mesentery for the stomach is likely to persist. And obviously the ventral mesentery has the liver in it, so it's going to persist. So there will be some, what do they call it? The adult structures, I'll use that term for the moment, or the remnants of these structures is another way of phrasing it, with new terms like the ventral, what is it called for the liver? Aha! I've forgotten what this is in the adult, so kick me in the butt. Okay, the orientation we have here also reminds us that structures are going to be growing and developing and pushing into certain areas, so they're going to distort that original, cylindrical shape of the pericardio-peritoneal canal. or cavity. And here, of course, it's now called peritoneal. We know we're in the abdominal cavity. So it will be deformed and reshaped as the embryo continues to grow and develop. Let's just take a moment to envision the lateral folds coming around to fuse in the ventral midline and form this new orientation of the peritoneal, or this peritoneal portion of the pericardio-peritoneal canal, such that we have a ventral and a dorsal mesentery. So what we have to envision with this butterfly wing again is that under each butterfly wing is a balloon. So it's one of the long cylindrical balloons that the clowns always work with. So it extends all the way up and curves in the front where the two wings would come together. We have to envision them coming together at the very front when they're wide open. And so as we push the wings down toward the front of the butterfly, the gut tube is developing there as well with some of its accessory structures, like we saw the liver here, and all the vasculature, the neurovascular bundle is developing at the same time. So these two balloons, those two ends of the same balloon, if you will, are going to be squashed onto those surfaces and will eventually come close to each other in the front and close to each other in the back. So if you want to consider taking just two balloons and a pencil in the middle, and you push the balloons against the pencil, ultimately the balloons are going to contact each other on each side of that pencil. So dorsally, where those balloons come together, is the dorsal mesentery, and ventrally is the ventral mesentery. So this holds true especially in the abdominal cavity, because it's almost clearly just lateral folds that create the abdominal cavity. Thoracic cavity forms with this pulling in of the U-shaped part of this balloon we're envisioning, pulling into the location ventral to the relative thoracic region. So we have slightly different arrangements of the body cavity spaces there. But for the abdominal pelvic cavity, it's sweeping in like this, and we're squashing two balloons together. Everything gets trapped in between them, unless it develops out here in the mesenchyme in the first place and was never suspended somewhere in the middle to be squashed between the two balloons. And so picture your fingers on the outside of the balloons as you're pushing them onto the pencil. Your fingers are always going to be outside the balloon. So there will be structures that develop like that, like the gonads and the kidneys and adrenal glands. So they are outside this layer, the peritoneum. So they're retroperitoneal. So it's going to become quite complicated as we proceed in all of these discussions. So therefore we see that there is still this discontinuity between the two separate portions or tails of this single balloon that we're envisioning. And in areas where the ventral mesentery is going to disappear, the peritoneal cavity is going to be continuous right across the front. And that will be true slightly farther caudally. But the gut tube has to complete its development. So obviously here we are seeing an area where the ventral fusion of the lateral folds has not completed its development, because it's surrounding the yolk sac, which is this extension of the midgut, middle portion of the gut tube. So we know that this is the body stalk region. So we start again first dorsal with the neural tube, the notochord, the dorsal aorta, and we recognize our two portions of the peritoneal cavity, the tails of that balloon that we've been using as a metaphor. And we have all of our orientation here. So ultimately, this has to become constricted to the point of becoming the attachment, of the umbilical cord, so that all of the ventral body wall fuses together. There are developmental anomalies where the ventral body wall doesn't fuse completely. The urinary bladder can be completely exposed on the surface and things like that, with incomplete fusion of the ventral body wall. Then as we continue to proceed caudally in panel E, We obviously are below the body stalk attachment. Dorsally neural tube, notochord, dorsal aorta. We see a dorsal mesentery for the hindgut region, and we're looking for that ventral mesentery. It's already disappeared by this time point, and so the peritoneal cavity is continuous on both sides of the organism. And we're beginning to see terminology being applied to the mesoderm lining the, or closely associated with the organs. There's two terms for organs: viscera, like the visceral layer of the peritoneum here, and splanchna, or splanchnic, of course, is the adjective for that. So visceral would be the adjective form, viscera would be the noun form, splanchna with an A there would be the noun form, and with the IC it's an adjective. So there's splanchnic mesoderm and somatic mesoderm. So soma obviously is referring to the body wall. The dorsal mesentery here is not showing it, but it would have contained within it either the branch, called the inferior mesenteric artery going straight in, or if we're not at that plane of section, there would be branches of the neurovascular bundle within the dorsal mesentery. This dorsal mesentery will not always persist. For example, if we were down where this is going to become the rectum, the dorsal mesentery would be lost on the rectum as well, because the rectum becomes plastered against the posterior body wall. So all throughout the gut tube, you'll have to know ultimately, did the dorsal mesentery persist? Did the ventral mesentery persist? If it didn't, how did it disappear? And what brought that on? And what is the terminology for the new structure? Thus, we review, in essence, the cavity membranes that line the structures. Pericardium is going to have the visceral layer on the organ itself and the parietal layer. The pleura, same thing, visceral and parietal, and we see that it contains common cardinal veins, so we're reminded that it's not just the arterial system that's present, it's the venous system, as well as the lymphatic system developing within here. We can't ignore that in our vascular bundle. And the nervous system is here as well. We've got the invasion of the branches, or migration of the neural crest cells to establish the sympathetic and parasympathetic innervation of these structures, as well as sensory fibers extending into there to contribute the sensory innervation of all of these organ systems we're considering. Here are the lungs, so we know there's stretch receptors, so there's got to be the receptor fibers coming in, the sensory fibers coming in and innervating the appropriate areas early on, staying with it to stretch and elongate to end up in the appropriate final location. So all of this is occurring as we're just tossing these simple terms out for us now. So we want to build the picture as best as we can when we are proceeding through these discussions. Well, now we want to take a look at an embryo of 24 days. And we see in panel A the lateral wall of the pericardial surface, the parietal pericardium has been removed so that we can look into the pericardial cavity and see the heart in its entirety here. We see now already that the heart has changed shape. It's in this constrained space, begins to elongate, and there's no place for it to go. It can't just push out and remain as a long cylindrical tube. So now we picture taking a linear piece of tubing and having it grow and twist, and ultimately it's going to twist around onto itself. And so the incoming and outgoing vessels are going to be in one end, and we've got all the chambers forming in there. So my daughter's taking a human anatomy course, and her teacher asked questions in a very strange way. Which muscle type has a figure 8 arrangement? I think we just saw the answer as cardiac muscle tissue, right? So it ends up with that type of twisting and contorting, so the cardiac myocytes form basically a figure 8 pattern surrounding the chambers. So if we take a section through this plane here at the level shown in panel A, we can take a look at that in panel B. And what it's beginning to show us is the development of the thoracoabdominal diaphragm. So we see the yolk stalk and the allantoic stalk. That immediately reminds us that's the body stalk, the primordium of the umbilical cord. So if we have a plane of section immediately rostral toward the head, or cranial toward the head, to that, we should have a plane of section that would leave the heart out, give us still our dorsal orientation, and let us see some of the mesoderm in that area as well as the nervous tissue. And we should see the primordium of the thoracoabdominal diaphragm. So we see that in panel B, and first let's just remind ourselves of the two balloons, two edges of the balloon. They're being pushed together and onto the developing structures within. And we go to our initial landmark, the neural tube dorsally, notochord. We see that there's two vessels here, so there's actually two parts of the dorsal aorta at this point. And the foregut is present there. We do not see any evidence of the lung buds developing, but we know it's going to push into the mesoderm, the mesenchyme in this area ultimately. And then we see the septum transversum, which is the primordium of the tendinous part of the diaphragm. So the diaphragm is the dome-shaped structure that we know. It's musculo-tendinous. And the central part of it is a tendon. Of course, at the periphery, there's tendons for insertion against the ribs and the body wall. So the central tendon is where these skeletal muscle fibers attach to their dense regular connective tissue, the tendons, and insert upon each other, basically at the central part or the dome of the diaphragm. So it's the septum transversum differentiates into this dense regular connective tissue layer. And the other mesoderm will come in and form the musculature, the skeletal muscle of the diaphragm, the thoracoabdominal diaphragm. So there's other mesoderm or mesenchyme in this area that's going to move in and surround the septum transversum, and all the cells will interact and the connective tissue will become continuous. Let's see here. At the body stalk, we have to remind ourselves, as we did in the prior series of sections, the panel that was right through the body stalk, that's the point where the ventral body wall has not completed its fusion yet. So the intraembryonic coelom and the extraembryonic coelom are still continuous at that point. And this, of course, reflects on the circulation of the amniotic fluid. Remember that the amnion has been pulled around at the body stalk as well, and is beginning to collapse onto the body stalk in its entirety. So this panel is just showing this continuity of the intraembryonic coelom with the extraembryonic coelom, and in the panel on the right, panel D, we can see this arrow pointing to the potential continuity of flow through this clown's balloon that we talked about in the first place. And also what we see present in this illustration is the front end of that bent balloon, where it's that U-shaped continuity, is now beginning to bend down and migrate into a ventral location. So right now, if we took a plane of section like we had before, through this area just rostral to the septum transversum, we'd see the developing heart, but we wouldn't necessarily see the pericardial cavity associated with it, because it has not migrated in completely to be associated with the heart in that area. Where this discussion leads probably would take us more than 2 minutes, so I'll just try to do an overview here and refer you to the online lectures that take you through the remaining series of slides. Is that the septum transversum, with the invading mesoderm, is going to create the separation between the thoracic and the abdominal cavity. The thoracic cavity with the pleural cavity, or the pericardial peritoneal cavity as it's called initially, and then we just start calling it the pleural cavity, is going to be reformed, reshaped by the ingrowth and development of structures and organ systems in that area. So this pericardial peritoneal canal is going to form as the bronchial buds begin to grow into this mesenchyme in the area. And the canal, I think I've used it erroneously earlier, the canal really refers to this narrowing of the cavity, because we have the pulmonary system beginning to develop in this area. As well, there are some folds that develop laterally because of these large veins, the left and the right cardinal vein, and I guess if they're calling this left, that blows my theory of looking from bottom down where right's right. And so apparently they're taking it and turning it around, where our right is the left of the organism, so we're looking from front down. As long as they label it left, we're okay, right? So the outpocketing of the developing lungs is not the only factor contributing to the narrowing of this cavity into the pericardio-peritoneal canal. And we see that this is even more extensive. This should be at a later time. B is at 5 weeks, and what's A? No, A is 5 weeks and B is 6 weeks, sorry. So within a week, this becomes much more well established, and we see the more characteristic appearance of the pleural cavity, and we see that the separation between the pleural cavity and the pericardial cavity is nearly completely established, as these lateral folds, the pleural pericardial membranes, are extending in and simply becoming thinner. So we can see that in this illustration from Gray's Anatomy, and I won't spend much time on this because we're a couple minutes over. But we see a mesentery already, the neural tube. So we know this is dorsal. We know the notochord would be there. Here's the 2 dorsal aorta that we had seen. And so there's a mesentery there if there's an attachment and a cavity nearby. So we see these lateral folds that we had mentioned as the duct of Cuvier. So we would not use this term, we would use the previous term from the previous slide to describe this. So the pericardio-peritoneal cavity is narrowed in this area, creating this canal, and eventually that canal is going to disappear with the fusion of these structures, the bulge created by the ingrowing lung and this lateral pleuropericardial membrane. So I'll stop there and express my appreciation for the extra couple minutes. And I'll see you on next Monday.